Yeah, so this story is actually a wonderful example of academia and the beautiful enterprise of being able to pursue anything you want. And this was an NIH-funded study, especially important in this climate these days. We couldn't have done this without NIH research. But essentially, we had a student that created one of the first CRISPR-Cas models for IDH wild-type mutant cells. We created this model and we did a high-throughput screen, and it was an unbiased screen. And we simply asked the question with really no preconceived notions: what molecules in the DNA repair and damage space would specifically target IDH mutations? We came into this really not knowing what we would expect, and to our surprise, we had a marked synthetic lethal interaction with PARP inhibitors. From there, the story really took off because we realized that this PARP inhibitor sensitivity was actually a signal that the IDH mutations induced an HR defect, essentially BRCAness. So these were no different than BRCA-deficient tumors, which really was coming out of left field. Really, many people were in disbelief when we reported this story. It took quite a time for people to really understand that this is truly what's happening with oncometabolites. Yep, exactly. So this is opening up not only a new area of treatment, and that's because we have FDA-approved PARP inhibitors that we can possibly test. This work is all in vivo and in vitro, and we're going to be beginning a trial soon, which we'll be speaking about at this conference here. But not only that, but it actually calls into question one of the main sort of treatment paradigms in place for these mutations. Our study specifically shows that oncometabolites actually drive this DNA repair defect, and that when you reverse them with many of the small molecule inhibitors of IDH1 and IDH2, you actually reverse the sensitivity. And we also will be presenting unpublished data today that the IDH1 and IDH2 inhibitors, at least in the laboratory setting, in our hands, in the hands of many of our collaborators, do not have the effects that have been described previously. So we really call into question the current clinical paradigm, and we really ask whether we need to actually consider exploiting the phenotype rather than suppressing it.